Hello everyone and welcome to today's session, and today we'll be discussing NCERT exercises for chapter Microbes in Human Welfare. This is the disclaimer. Okay, so in today's session we will understand the basic concepts related to the chapter Microbes in Human Welfare by working through the NCERT exercises questions. So these questions, they are based on certain concepts. We will understand these concepts by discussing these questions. Okay, so we'll be discussing NCERT exercises for the chapter 8, which is Microbes in Human Welfare. Okay, let's begin with the first question. The first question is, bacteria cannot be seen with naked eyes, but these can be seen with the help of microscope. If you have to carry a sample from your home to your biology laboratory to demonstrate the presence of microbes with the help of a microscope, which sample would you carry and why? For more information, you can read NCERT section 8.1. So let's have a look at the answer. Okay. So curd is an excellent example for demonstrating bacteria under a microscope. Okay. So under the microscope, you can easily observe bacteria. Clear? So a sample of curd would be ideal to carry to the biology lab for microscope observation of the bacteria. Okay. Now, a tiny drop of curd has millions of lactobacillus bacteria, which means there are plenty of bacteria to observe. If you observe curd under a microscope, this helps us to connect the theoretical knowledge with the practical observation. Now, these bacteria can be easily seen and identified under a microscope, making them great for demonstrations. Okay, so curd is quite safe also. Okay, it is safe to handle and can be easily found at home. So it's very convenient choice for lab sample. So here you can see, in this image you can see lactic acid bacteria, okay, which helps in curd formation. Next question, give examples to prove that microbes release gas during the metabolism. So let's have a look at the explanation. See, when microorganisms like bacteria and yeast, they break down sugars, they often produce gases as the byproducts. Okay, a well-known example of this process is fermentation of dough. Okay, used for making food like idlis, dosas, and bread. And during fermentation, carbon dioxide gas is produced. Okay, what is fermentation? Anaerobic respiration. So this gas causes the dough to rise, giving it light and a fluffy texture. Okay, now the puffy look of the dough. This is due to the carbon dioxide released by the microorganisms during the fermentation. Okay. Now, baker's yeast, also known as Saccharomyces cerevisiae. So, yeast scientific name is Saccharomyces cerevisiae. Okay, this ferments the sugar present in the dough, producing carbon dioxide that makes the bread rise. So, you can see here glucose, which is simple form of carbohydrate. This is broken down in the presence of oxygen producing energy and carbon dioxide. Okay, in absence of you can see oxygen, glucose is converted into alcohol and carbon dioxide. The next question, in which food would you find lactic acid bacteria, and you have to mention some of the useful applications. Okay, so let's have a look at the explanation. So lactic acid bacteria are commonly found in following fermented dairy products. Okay, what are these products? First is yogurt. Okay, so these bacteria, they are, you can see, beneficial also. They are vital allies, they are promoting health, and they're enhancing the food quality also. Like lactic acid bacteria, it enhances the vitamin B12 content in yogurt. Okay, then you have curd. So there is a difference between curd and yogurt. In curd, you have more number of microorganisms. In yogurt, you have more number of microorganisms. You have mixture of microorganisms in yogurt. In curd, you have lactic acid bacteria. Then you have buttermilk. Okay, so you can see, okay, these are the food which commonly use lactic acid bacteria. These are yogurt, curd, and buttermilk. Okay, now useful application of lactic acid bacteria. Okay, so it enhances the nutritional value of the food. So lactic acid bacteria can increase the nutrition value of food by enhancing the vitamin B12 content in the curd. Okay. Next, it maintains gut health as well. Okay, so lactic acid bacteria are beneficial for human gut health, helping to maintain a healthy balance of the bacteria. Then they help maintain a healthy balance of bacteria in the digestive system and can inhibit the growth of harmful bacteria as well. Okay, again you can see the morphology of the lactic acid bacteria as well here. So these bacteria are used for the preparation of yogurt and curd and buttermilk as well. Next, name some traditional Indian foods made of wheat, rice and Bengal gram. Okay, or their products which involve use of the microbes. Okay, so you can see the table here, the material and the products are given to you. You can see wheat. This is used to make bread and cake. Then rice, this is used to make idli and dosa. And then Bengal gram, this is used to make dokla and khandvi. Yeah, so this was a very simple and straightforward question. In this image, you can see bread, dokla and idli and dosa. These are traditional Indian foods made up of wheat, rice and Bengal gram. Next question, in which way have microbes played a major role in controlling diseases caused by harmful bacteria? Okay, so let's have a look at the answer. See, these microorganisms, they help fight diseases by making substances or by producing substances, by secreting substances, by secreting certain biomolecules, okay, which are known as antibiotics. Clear? Or by secreting certain, you can say, secondary metabolites. Okay, so these, some of them are antibiotics. They can kill or they can stop the growth of harmful bacteria. Now, these antibiotics, often derived from bacteria and fungi, either kill or inhibit the growth of disease-causing bacteria. Okay, so either they kill or they slow down the growth of the bacteria. Okay, example is penicillin. This is antibacterial. This is produced by the fungus Penicillium notatum. Okay, what is the mechanism of action? It weakens the bacterial cell walls. Okay, clear? Okay, or you can say it causes the breakdown of the bacterial cell wall. It makes bacteria vulnerable to the immune system. So penicillin attack and weaken the protective outer layer of the bacteria. Okay, so that's why it's become more vulnerable to the cells of the immune system. Okay, now when the cell wall is destroyed or weakened, the bacteria become, you can say, more vulnerable to the immune system attack. Okay, so we can say these antibiotic, they help body destroy the harmful bacteria. So here you can see in this image, you can see penicillin mold, and they have magnified also. You can see the penicillin colony here. and how the mycelia looks. Next question, name any two species of fungus which are used in the production of the antibiotics. So let's have a look at the answer C. Okay, what is antibiotic? Let's understand this word. So anti means against, bio means life, means against life. Okay, we are talking, you can say about this in the context of disease causing organisms. So they are, you can say, killing, or they are leading to the, or they are causing slow growth of the microorganisms. Okay, so they are against life, but in context of disease causing organisms. Okay, so these are basically chemical substances which are produced by some microorganisms, and they are commonly obtained from bacteria and fungi. Okay, so these bacteria and fungi, they produce large amount of secondary metabolites. Okay, some of them are bioactive. They have bioactivity. Okay, so these can be antibacterial, anti-inflammatory, clear, and so on. Now the chemicals can kill or they can retard the growth of other disease-causing microorganisms. Okay. Now the species of fungus used in production of antibiotics are, okay, so antibiotics are penicillin and cephalosporin. Okay, so source of penicillin, Penicillium notatum, and source of cephalosporin is Cephalosporium acrimonium. Next question, what is sewage and in which way can sewage be harmful to us? Okay, so let's have a look at the answer. Okay, so sewage is the waste from homes and from other areas that is removed through pipes called sewers and the drains. Okay, basically it is the municipal waste matter that is carried away in the sewer and the drains. Okay, so this sewage contains both liquid waste and solid waste, and it has a lot of natural materials also. Okay, organic matter also, and tiny living, you can say, organisms called microorganisms as well. Okay, so it includes both liquid and solid waste rich in organic matter and microbes. Now sewage can be harmful to us because, okay, now some of the microbes in sewage can make us sick because they are harmful and can lead to the discussion that spread through the water. Okay, so many of these microbes which are present in the sewage, they are pathogenic, they can cause disease, and they can cause several waterborne disease as well. Okay, so they can cause disease, they can you can say lead to disease that can spread through water. Okay, now when sewage mixes with water sources, it can contaminate drinking water as well, making it unsafe to drink. The next question, what is the key difference between primary and secondary sewage treatment? Okay, so let's have a look at the answer. See, this is a table showing you difference between primary sewage treatment and secondary sewage treatment. Okay, so what happens in primary sewage treatment? It is a mechanical process involving the removal of coarse solid materials. Okay, it is inexpensive and relatively less complicated. And secondary sewage treatment, it is a biological process involving the action of the microorganisms. It is very expensive and complicated process. Okay, in this image you can see primary treatment and the secondary treatment. Okay, clear? So here you can see in the primary treatment there is the removal of coarse solid materials. Okay, so it has a grit chamber you can see, and then a sedimentation tank as well. Clear? And in the biological, this secondary sewage treatment, it is basically biological process. It involves microorganisms. Next question, do you think microbes can also be used as a source of energy? If yes, how? So let's have a look at the answer. Okay, so yes, microbes can be used as a source of energy. Okay, the bacteria such as methane bacterium is used for the generation of gobar gas or the biogas. Okay, so basically methane bacterium is methanogens. They are used for the production of biogas, which is used as a biofuel. So methanogens, these are basically anaerobic bacteria. So they are involved in biogas production. What is the process of biogas production? So some special microbes known as methanogens, they break down the organic matter, which is rich in cellulose, okay, in the absence of the oxygen. Now, the organic materials that these microorganisms break down can be things like animal waste. Okay, so organic matter includes animal manure or sewage. Clear? So here you can see this is the image showing you importance of the biogas. So biogas can be used as a fuel. It has various applications, and then the you can say spent slurry, the leftover slurry, this can be used as a biofertilizer as well. Okay, so let's move on. Okay, we're talking about the biogas production mechanism. Okay, what is the mechanism? So these bacteria, they break down the organic matter into simpler compounds. Okay, now as the bacteria do their work, what is happening here? The biogas is being produced, which is rich in methane. What is the application of biogas? So this methane rich biogas can then be collected and used as a fuel source for cooking, for heating, for electricity generation as well. Okay, so it can be used as a cooking, so it is quite inflammable. That's why it can be used as a cooking. It generates energy. It can generate heat. It can even be used for generating electricity. Again, you can see this is an image showing you various application of the biogas. Next question, microbes can be used to decrease the use of chemical fertilizers and pesticides. You have to explain how this can be accomplished. Okay, so microorganisms can significantly reduce the need for chemical fertilizers and pesticides through the use of biofertilizers and biopesticides. Okay, let's understand this. First of all, let's talk about the biofertilizers. Okay, so biofertilizers, these are basically microorganisms such as bacteria, fungi, and blue-green algae. Okay, these microorganisms, they can fix atmospheric nitrogen. Okay, so what happens in nitrogen fixation? So these bacteria, they can convert atmospheric nitrogen into forms that the plant can use. Okay, clear? So many bacteria can convert atmospheric nitrogen into usable forms for the plant. Now this reduces the need for nitrogen-based chemical fertilizers. Okay, these chemical fertilizers, they are non-biodegradable. Okay, they accumulate in the environment, they degrade the environment, and they ultimately enter in the food chain also. Okay, now bacteria like Rhizobium, Azotobacter, and Azospirillum, they can do nitrogen fixation. Okay, cyanobacteria like Anabina and Nostoc, this can also carry out nitrogen fixation. So you can see this is an image showing you Rhizobium, which is a bacteria which lives in the roots of the leguminous plants, and it forms root nodules where it carries out nitrogen fixation. Okay, second is biopesticides. Okay, so they targeted pest control can be done. So biopesticide, they are effective against specific pests like the insects. Okay, so they are insect group specific. Okay, they pose minimal harm to the beneficial insects or humans and other animals. Okay, so they are not harming any other species, any other organism which are present in that area, in that environment, or in that ecosystem. They are targeting a specific pest. Examples: Bacillus thuringiensis bacteria. So this bacteria produce Bt toxin. Okay, this Bt toxin, when it enters into the insect's gut, then it gets activated due to the alkaline pH of the gut, and then it creates holes in insect's gut and the insect dies. So first one is Bacillus thuringiensis. Importance is it is a toxin that kills the insect pest. And second is baculoviruses. It is used as a biological control agent against insects and other arthropods as well. Okay, you can see this is an image showing you Bacillus thuringiensis, which is a soil bacteria, and this bacteria produces Bt toxin crystals. So this bacteria produces toxin in inactive form, which gets activated in the insect's gut. And this gene, Bt gene, also known as cry gene, has been introduced into crop plants such as Bt cotton. Okay, what is the significance of biofertilizers and biopesticides? Okay, first of all, reduced chemical residues. So biofertilizers, basically they are organisms such as bacteria, fungi, and blue-green algae, okay, cyanobacteria, that enrich the nutrient quality of the soil. Okay, they enhance the soil fertility as well, and they reduce our dependency on the chemical fertilizers. Now, biopesticide offer a natural way to control the pest, minimizing the reliance on the harmful chemicals. Next question: 3 water samples, namely river water, untreated sewage water, and secondary effluent discharge from a sewage treatment plant, were subjected to BOD test. The samples were labeled A, B, and C, but the laboratory attendant did not note which was which. Now, the BOD values of the 3 samples A, B, C were recorded as 20 mg per liter, 8 mg per liter, and 400 mg per liter, respectively. Which sample of the water is most polluted? Can you assign the correct label to each, assuming the river water is relatively clean? Okay, so let's have a look at the answers. See, let's talk about biological oxygen demand or biochemical oxygen demand. Okay, so what is BOD? It is a measure of the organic matter present in the water. Okay, now the amount of oxygen required for oxidizing all the organic matter in 1 liter of water by bacteria, this is known as BOD. Now, high BOD indicates high pollution potential, means there is more organic waste in the water. There is more organic matter in the water. Low BOD indicates cleaner water. Okay, in clean water there is less organic matter, so its BOD will be less. The amount of oxygen which is used by the bacteria to degrade this organic matter, this is basically BOD in 1 liter of water. Okay. Now, from the concept of BOD, following conclusions can be drawn. So it can be concluded that if the water supply is more polluted, then it will have a higher BOD value. Okay. So more pollution, higher BOD. Now, out of the above 3 samples, sample C is the most polluted since it has the maximum BOD value of 400 mg per liter. Now, after untreated sewage water, secondary effluent discharge from a sewage treatment plant is most polluted. Okay, the sample A is secondary effluent discharge from the sewage treatment plant and has the BOD value of 20 mg per liter. The sample B in river water, okay, and has the BOD value of 8 mg per liter. Now the correct label for each sample is, okay, so you have the label A, B, C, BOD value is 20 mg per liter for A, 8 mg per liter for B, 400 mg per liter for C. Okay, for A, okay, the sample is secondary effluent discharge from a sewage treatment plant. Then B, this is the river water. Okay, then C, this is the untreated sewage waste. Okay, this is clear. So let's begin with the next question. So you have to find out the name of the microbes from which cyclosporin A, an immunosuppressive drug, and statin, which is an blood cholesterol lowering agents, are obtained. So let's have a look at the answer. Okay, so cyclosporin A, which is an immunosuppressive drug, is obtained from Trichoderma polysporum. And statin, which is a blood cholesterol lowering agent, is obtained from Monascus purpureus. The next question, you have to find out the role of microbes in following and discuss it with your teacher. So A is single cell protein and B is soil. Let's have a look at the answer. Okay, so single cell protein. How can you define single cell protein? So single cell protein refers to the dried cells of microorganisms. Okay, these microorganisms can be algae, bacteria, fungi, or yeast. Okay, so these are rich in protein and used as a food for or the feed supplement. Okay, what is the microbial role here? Now, the microbes involved in the preparation of single cell proteins are, okay, so microorganisms like spirulina, which is algae. Okay, now these are involved in the preparation of the single cell protein. Okay, or these single cell proteins are obtained from spirulina. Here now, methylophilus. Okay, methylotrophus, which is a bacteria. This is also used for the preparation of single cell proteins. Then various yeasts and fungi are cultivated on a large scale to produce single cell proteins. Okay, let's continue with the answer. What are the benefits of single cell protein? It serves as a rich supplement of dietary nutrients such as protein, carbohydrate, fats, minerals, and vitamins. Single cell protein can help address food security issues by providing an alternative protein source, especially in regions with protein deficiencies. Examples. Okay, so spirulina is a well-known example of single cell protein source. This is because of its high protein content and ability to be cultivated easily. So spirulina is a superfood. It is very nutritious. Clear? Here you can see in this image single cell proteins which are obtained from spirulina. Clear? Let's move on to the next question. Now, let's talk about soil. So microbes play an important role in maintaining soil fertility. They help in the formation of nutrient-rich humus by the process of decomposition. Many species of bacteria and cyanobacteria have the ability to fix atmospheric nitrogen into usable forms. Okay, clear? So they can fix atmospheric nitrogen and they can convert nitrogen into a form that can be absorbed by the plants. Okay, so Rhizobium, this is a symbiotic bacteria. This is found in the root nodules of the leguminous plants. Okay, it is involved in atmospheric nitrogen fixation. Examples. Azoospermillum and Azotobacter, these are free-living nitrogen-fixing bacteria. So bacteria can form symbiotic association or they can be free-living, like Azoospermillum and Azotobacter. Now, Anabina, Nostoc, and Oscillatoria, they are examples of nitrogen-fixing cyanobacteria, which is also called blue-green algae. Okay, so here you can see in this image Rhizobium and cyanobacteria. So Rhizobium, it you can say it formed association with the roots of the leguminous plant. Clear? And they are involved in atmospheric nitrogen fixation. Next question, you have to arrange the following in the decreasing order. Most important first of their importance for the welfare of human society. You have to give reasons for your answer. Biogas, citric acid, penicillin, and curd. Let's have a look at the answer. Okay, the order of arrangement of products according to their decreasing importance is penicillin first, then biogas, citric acid, and then fourth one is curd. So penicillin. Penicillin is the most important product for the welfare of human society. So it is an antibiotic obtained from the fungus Penicillium. This antibiotic is used for controlling various bacterial diseases. Second is biogas. It is second most popular product. Second most important product is biogas. It is an eco-friendly source of energy. Okay, so biogas can be used for lighting. Okay, can be used for electricity production as well. Clear? It can be used for cooking as well because it is inflammable. Citric acid. The next important product is citric acid, which is used for food, used as a food preservative. Then curd. It is the least important product. Okay, this is a food item obtained by the action of lactobacillus bacteria on the milk. Next question, how do biofertilizers enrich the fertility of the soil? Okay, so let's have a look at the answer. See, biofertilizers are basically living organisms that enrich the nutrient quality of the soil. Okay, so these biofertilizers can reduce the dependency on the chemical fertilizer which are toxic. They degrade the environment. These microorganisms which are used as biofertilizers, they play a crucial role in fixing atmospheric nitrogen, solubilizing phosphorus. Okay, so some of the biofertilizers such as mycorrhiza, they can solubilize phosphorus. Okay, and some of these microorganisms, okay, such as bacteria, they can fix atmospheric nitrogen as well. So first is nitrogen fixation. Certain bacteria can convert atmospheric nitrogen into ammonia. This is known as atmospheric nitrogen fixation. Okay. So atmospheric nitrogen is converted into ammonia, a form usable by the plants. Okay. Okay, example is rhizobium. This forms symbiotic association with the roots of the leguminous plant. So rhizobium can carry out atmospheric nitrogen fixation where atmospheric nitrogen is converted into ammonia. Okay, then ammonia is further converted into nitrites and nitrates. Okay, it forms symbiotic relationship with the legume plants. It lives in their root nodules and supply nitrogen directly to the plant. This is an image showing you rhizobium, which is living in the root nodules of the leguminous plant. Okay, then next. Now bacteria can be free living also. These free living bacteria, they can also fix atmospheric nitrogen. Okay, making nitrogen available for the plants. Example is Azoospermillum and Azotobacter. Now cyanobacteria can also fix nitrogen. Okay, particularly in aquatic and paddy fields environment. Examples are Anabina and Nostoc. So these cyanobacteria, they have heterocysts in which they carry out nitrogen fixation. In this image, you can even see heterocyst in the cyanobacteria. Okay, second is phosphorus solubilization. So some fungi like the genus Glomus, which form mycorrhizal association. Okay,
So mycorrhizal association is a symbiotic association with the roots of the plant. So these fungi help solubilize phosphorus, making it more accessible to plants from the soil. So plants cannot directly absorb this phosphorus from the soil. So this phosphorus is first solubilized by this fungus, and this phosphorus is converted into a form which can be absorbed by the plants. Okay. So this is particularly important as phosphorus is often present in insoluble forms in the soil. So plants cannot absorb, okay, this insoluble form of the phosphorus. So that's why phosphorus is solubilized first. In this image, you can see mycorrhizal association. Okay. So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.